Good afternoon. Thank you for joining us. And I have 4 minutes after the hour. Let's get started so we have time for discussion. Today is our first officially Grand Rounds of ENACT. It's a pleasure to have you here in inaugural session. And we'll be talking about trends in postpartum hemorrhage prevalence and comorbidity burden among women, insights from the ENACT network. I am Nayeli Mora, and I'm going to be moderating the session. I'm a research associate professor from Loyola. I am part of the dissemination and evaluation group from ENACT. As a reminder, ENACT network is it's a platform of electronic health record network and based in the CTSA consortium, academic research institutions, and it covers over 142 million patients. It evolves from the ACT core discovery platform with many other tools that you can see that enable us to get our data quality, access to trainings and educational purposes. We have the enclaves, which we've been talking about today. We're going to have other features that NLP and will support clinical decision making. And the purpose of Grand Rounds is that these monthly seminars, we are open to everybody with regardless of the membership to the CTSA, and we would just want to discuss what can we be doing and leveraging electronic health record networks, especially in ENACT, and what projects and case uses can we have with this platform. Again, it's a focus on open science, data sharing, collaboration, and our purpose is to have these interactive sessions with presentations, panels, networking, and a lot of discussion. So we encourage you to put your questions in the chat as we go. This is an overview of our session. And our first speakers today is Dr. Shyam Visweswaran and Dr. Olga Kravchenko. Dr. Shyam Visweswaran is a professor of biomedical informatics and vice chair of clinical informatics at the University of Pittsburgh. He's the director of the Center of Clinical Artificial Intelligence in the Department of Biomedical Informatics. His research interests include AI-enabled clinical decision support, racial equity, and clinical algorithms, patient-specific modeling, and large-scale EHR data analysis. He is one of the PDs and PIs of the ENACT network and oversees the informatics activities. Dr. Olga Kravchenko is an assistant professor of biomedical informatics at the University of Pittsburgh. Her research interests include developing practical AI, ML, and statistical models that can be translated into clinical settings, creating analytical tools that inform clinical decision support using EHR data, and translational research using large-scale EHR datasets. She's part of the enclaves group and the clinical evidence group in the ENACT network. And together they're gonna present to us how we can leverage the enclaves. A refresher on how to understand ENACT for real-world data analysis, to learn about a specific study that was used, aggregated counts for ENACT, and then has processed to using the ENACT enclave environment for further analysis. We also want to learn how to find additional information on the enclaves and what is needed actually to participate in these projects. With any further ado, Dr. Visweswaran,
Thank you, Nelly. So I'm going to start off by giving you an overview of ENACT and especially the enclaves, and then hand it over to Olga to talk about the postpartum hemorrhage study. So as most of you probably know, it's a federated network which started out as ACT and now it's called ENACT. And it links most of the CTSAs across the nation, and we can access a large volume of EHR data, especially structured EHR data. So the unique feature of this network is that any investigator at any of the participating sites can basically log on to a Shrine interface tool and query the network interactively. So they can come up with any question, convert it into a query using structured terminology, and that gets sent across the network, and the counts of patients who satisfy that query then come back. And so that's sort of where the whole network started, and that's the fundamental technology platform that we have in this network. One thing I want to point out is that when the network started up, the technology required that every site needed to have an I2B2 data repository. And now we have the ability to actually link up OMOP data repository. And the network works seamlessly with the other data model, which is OMOP, and eventually we'll have the PicoNet CDM also be able to be interoperable. So this makes it relatively easy to, if a site wants to participate with a different data model other than I2B2, especially OMOP, to connect up to the network. And this is just to show that the amount of work that you have to do if you are having an OMOP data repository is not that much, that you still have to install the software, which includes I2B2 and the Shrine and the ontology, but the data itself doesn't have to be sent to the I2B2. And with that, you should be able to interoperate with the network. So as we moved from ACT to ENACT, ACT was set up to do cohort discovery. And with ENACT, we moved into doing research on this platform. And there's three broad kinds of analytics that you can do are aggregated patient counts, ephemeral enclaves, and distributed analytics. And in this session, you will see how we have used aggregated counts as well as ephemeral enclaves. So aggregated counts basically uses the initial ACT technology that we set up, which uses Shrine and gives the ability for an investigator to do interactive querying of the network and obtain patient counts. And even with aggregated patient counts, you can do a fair amount of research. And ephemeral enclaves enables you to do even deeper research compared to aggregated counts. So this is just a snapshot of the current Shrine interface. And just of note, the new I2B2 also looks like the Shrine interface. So these are the modern interfaces that these tools have. And you can run queries, which are relatively easy to do. And within minutes, you get counts back. And right now, the way the platform works is that we have obfuscation set up to this plus or minus 10 patients. And over the summer, when we upgrade the network, that obfuscation is going to get reduced to plus or minus 3, and that will enable us to do these counts-based research even for very rare conditions. Right now, if a site has 10 patients or less for a query, it's going to return as 10 or less. And so for rare conditions, you may not be able to actually get the actual counts that you want. So over the past 6 months, we have enabled ephemeral enclaves in this network. And this is basically the ability to move data from the local data repositories for a particular project to a central AWS platform. And then that enables investigators to do research. And the way it is set up in ENACT is that you still have to use Shrine to develop the cohort query. And this enables every site which contributes data to have the same definition for that cohort. And once that query has been developed and transmitted across the network, then the sites which participate in the study will move the data corresponding to the cohort to the AWS enclave. And once it's moved there, study investigators can log in and participate in the research. And you can have multiple investigators from multiple sites who will be able to log into that enclave and do their research. Right now, the enclaves are maintained by University of California at San Diego, and they have a technical team which helps enabling the account, making sure that network access is obtained, and so on and so forth in case there are technical issues. One unique feature of our enclaves is that after a study is completed, the enclave is archived, or the data is archived. So basically, the enclaves are set up to do studies which are specific to a particular cohort. And after the study gets done, the data is archived. So for every study, basically, a new cohort is set up and a new enclave is set up for that particular study. And the environment has got all the typical packages in Python and R. And typically, you do need the ability to write programs and code to be able to do analysis in this environment. And to some extent, it's similar to the environments which are available in the All of Us program and in the N3C program. So we have about half a dozen pilot projects currently in progress in the enclave. These are two examples. One is EHR-based predictive modeling of postpartum hemorrhage, which towards the end of the session, I'll talk briefly about. Here, the objective is to develop and validate machine learning model to predict postpartum hemorrhage. The second one, which I'll just briefly touch here, is predicting progression to end-stage renal disease in chronic kidney disease patients. Here, the objective is to validate a previously developed machine learning model, which was developed here at Pitt, that predicts the risk of early progression to end-stage renal disease in kidney patients on data from other institutions. And then with that, I'll hand it over to Olga to describe the PPH study.
Thank you, Shyam. Hi all. So I'm going to talk about our study that we in the study we used ENACT network to look into trends and comorbidities among women who experienced a complication known as postpartum hemorrhage after giving birth. And our goal here was to explore the use of ENACT as a research tool in practice. And we wanted to establish a process for working with this tool and to identify potential challenges or limitations that researchers might encounter while doing research in ENACT. We focused on postpartum hemorrhage, or PPH, as a use case for two reasons. One is that it is a significant clinical challenge, and the other reason is that we could use findings from a prior study that used a nationwide inpatient sample. And that study, it showed an increase in postpartum hemorrhage cases between years 2000 and 2019 in the United States. And the study also used a phenotype that was a well-defined phenotype, set of diagnostic and procedure codes, and we could apply this phenotype to our study as well. So we had something to compare our results to. We have used the process that was described earlier in this talk to extract the data from ENACT and to conduct our analysis. Now, PPH is a serious condition. It is defined as excessive blood loss within 24 hours of childbirth. It is associated with several maternal risk factors, such as advanced maternal age, obesity, multiple gestation, which is having twins or triplets, a history of postpartum hemorrhage, prior uterine surgery, pregnancy complications such as preeclampsia, labor induction, macrosomia, and emergency cesarean section. Again, the main objective was to examine trends in PPH and comorbidities between the years 2005 through 2022. This was the data that was available to us. And we also wanted to see if our results are consistent with previously published data that used an AS sample. And we wanted to explore whether this is a good resource for maternal health research. And overall, we wanted to validate Shrine as a research tool. We conducted repeated annual cross-section analysis using data that spanned 18 years from 2005 through 2022. We started by identifying unique individuals who had delivery hospitalizations in each calendar year within this time period. And then within this cohort, we identified deliveries with PPH using ICD-9, ICD-10 codes. And women with delivery hospitalizations without PPH served as a comparator group. Now, when we submitted our queries, we had two query sequences, one for all deliveries, and the other one is deliveries with PPH diagnosis. So we had two parallel streams of queries. And in both of these streams, we submitted queries for deliveries with and without PPH, each comorbidity and risk factor with and without PPH, demographics with and without PPH, and we did it for each year of the 18-year period. Now, when the query is submitted in ENACT, it goes to all contributing sites simultaneously. And there are over 50 contributing sites in ENACT. And for each separate query, we saw about half of them return results. So after we extracted this data, we had a set of inclusion criteria that we applied to the sites. Our starting cohort included sites that had no more than 50% of missing annual data, and this was our initial criteria. And this is equivalent of 9 years of missing data for the 18-year period, or those points that showed values significantly lower than adjacent years by at least 2 orders of magnitude. And this could indicate that this is a result of obfuscation or there's some missing EHR records for this year. We also excluded data from 2023 because at the time of extraction, contribution for this year was not complete by all health sites. And eventually we chose 13 sites that responded to all queries that we ran and satisfied the inclusion criteria. And this was later reduced to 8 sites after we conducted sensitivity analysis to determine missing values threshold. Our primary outcome measure was annual PPH incidence rate among delivery hospitalizations. And we also wanted to compare demographics, comorbidities, and risk factors in the group of women who had PPH after delivery and those who did not. And we have looked at race and various comorbidities and risk factors. For statistical tests, we used Mann-Kendall test to evaluate temporal trends and PPH incidence. We used this test because, well, this test is used to identify if there is a monotonic upward or downward trend over time. And we looked at trends by race, comorbidities, and maternal risk factors. And we also looked at prevalence of comorbidities among those who had PPH and prevalence of PPH within those groups of individuals who had these comorbidities. Our initial analysis focused on assessing or data quality analysis, I would say. So we focused on assessing the extent of missing data in the extracted data set. And we wanted to determine an appropriate threshold for missing values. So first, we calculated the postpartum hemorrhage incidence rates for sites with no missing values to establish a baseline trend. And then we grouped contributing sites based on the percentage of missing data into 4 categories: 0%, which is a baseline, less than 10%, less than 20%, and less than 50% of missing data. And then for each group, we imputed missing values. Either we used linear regression when there were 2 or more consecutive missing values, missing years, or for a boundary year. And for single year missing data, the counts of the preceding and subsequent years were averaged. And then we calculated the annual incidence rate for each group, and we compared these rates with the baseline data. And we used Pearson correlation coefficient between the pairs of normalized curves. And as a result of this analysis, we chose that the group with less than 20% missing value would be appropriate in the study. This would maximize inclusion of data, but it wouldn't compromise data quality too much. And this reduced the number of sites in all further analysis to 8. So I will show some of the results. The full set of results is available in our preprint that is online, and there will be QR code in the, on the last slide. And here is the data for racial subgroups. The data, we have data for total deliveries, PPH deliveries, and non-PPH deliveries. Here the data is presented in raw numbers and in colon percent. And the colon percent shows the share of a demographic subgroup in these different, in total deliveries, and PPH deliveries, and non-PPH deliveries. And the reason to do it this way is that we can compare characteristic distributions of these subgroups between those with PPH deliveries and those without. So here you can see that in the total deliveries column that deliveries, that the largest share of deliveries in our cohort was among white mothers, They accounted for about 67% of all births. And the smallest subgroup is a group of native Hawaiian or other Pacific Islander mothers who accounted for 0.23% of all births. So in the next two columns, the white mothers was the only group that had lower share of PPH deliveries than in non-PPH deliveries, and all other groups had higher share in PPH deliveries than in non-PPH deliveries, except for multiple race subgroup and no information subgroup. And we used chi-square test to compare representation in these subgroups. Another way to present this result is by looking at the absolute risk for each subgroup or incidence rate of PPH in each subgroup. And here we can see that native Hawaiian or other Pacific Islander group has the highest incidence rate, and white women subgroup has the lowest PPH rate. And if we convert it to crude incidence ratio and use white women subgroup as a baseline, then we can see that the relative risk for all other subgroups increases compared to white women subgroup. For example, the relative risk for Black or African American women to experience PPH is 13% higher than that for white women. Now, this slide shows the absolute risk of postpartum hemorrhage increases on how the absolute risk increases as the number of risk factors or comorbidities accumulates. Deliveries with no identified PPH risk factors had an absolute risk of 5.35%. And then for those with at least one risk factor, the risk jumps to 12.7% and goes up further to 13.4%, and those with at least two risk factors. And a similar pattern is, we can see that there is a similar pattern for comorbidities. PPH risk is 6.75% in deliveries with no comorbidities, and it increases to 11.9 with at least one, and reaches 15% in those with two or more comorbidities. We also looked at the breakdown of risk factors and comorbidities, and this figure shows the distribution of these factors. Maternal risk factors as blue bars, and comorbidities as red bars, and race and ethnicity as green bars. And we discussed them in previous slides. The bars represent the proportion of PPH cases associated with each factor. So for comorbidities, we found that among women who experienced PPH, antepartum hemorrhage, and placental abruption, as well as vaginal operative deliveries, were the most common comorbidities. And for risk factors, we found that placenta previa creta, florium, amnionitis, endometritis, and severe preeclampsia are among the most common risk factors. And this figure shows the trend in the PPH incidence rate for years 2005 through 2022. And the increase was from approximately 5,600 cases to about 10,500 PPH cases per 100,000 deliveries. And this trend remained consistent among people with more than one comorbid conditions and those with more than one maternal risk factors. And I'm not showing these plots, but we report them in our paper that you can find online. This trend is broadly consistent with the previously reported trend that showed data from 2000 through 2019. In our study, the trend is a little bit steeper, but it also doesn't have data from prior to 2005, and it extends beyond 2019. And we can see that it continues to increase after 2019, and we can also see that it is even steeper probably in the last few years. And our study had some limitations. So one of the limitations was that we could not use age because when you extract age in ENACT, it is the age at the point of extraction, so the current age of a person. We could not derive the age at the delivery or age at the diagnosis. Another limitation is that the patient counts that are provided in Shrine are obfuscated. This is done to preserve patient privacy, and this may introduce some level of inaccuracy. And the counts are obfuscated in such a way that they're rounded to the nearest 5, and then they're further obfuscated by adding or subtracting up to 10 counts. And this means that when you rerun your query, you may see slightly different results. And these variations are typically very small. And if you're working with large counts, this is not a problem. But you have to be very careful if you work with smaller counts and examine whether the counts that you have, whether they can be an artifact of obfuscation, or whether they're so small that obfuscation makes your results inconclusive. Another limitation is that the technology used in ENACT calculates the number of unique patients that meet a query criteria, and this may lead to undercounting some patients who had multiple occurrences of the same diagnostic code. For example, if there are multiple deliveries, they can be undercounted. But in this case, we mitigated this by setting a time constraint of 1 year to obtain unique counts, because the pregnancy lasts 9 months. And the mitigation strategy, it depends on your clinical question at hand. Another limitation, we used ICD-9, ICD-10 codes only. We did it because our phenotype was based on previous methodologies. We did not use CPT codes, but in principle, this could lead to incomplete data capture, and we can potentially miss some delivery procedures. And of course, we cannot follow individuals longitudinally in this type of analysis, and we are limited by the Shrine constraints and how many criteria we can implement in a query. So criteria can be very sophisticated, but there are limits to this. And despite all of these limitations, the data obtained in Shrine is a great source of real-world evidence that you can obtain very fast, and that can be used in a number of different problems. And this is the team that worked on the study. Thank you to the team. And here is a QR code that leads to the preprint. Our paper is currently under review, but there are more details in there that you can read. And thank you very much. I will pass it to Sean.
Yeah, thank you, Olga. So Olga presented how you can use counts using the Shrine tool to do fairly sophisticated analysis. And some of the limitations she pointed out, one of them was the obfuscation problem, where right now it's set up to plus minus 10. Over the summer, we're going to be rolling out a much lower threshold. It's going to be plus or minus 3. And so that should enable even doing research on conditions which have somewhat smaller counts. So usually when you end up doing this kind of counts-based research, you end up with probably having even more questions that you want to work on, which involves using patient-level data. And so that's kind of what happened with this project too, is that we see that the temporal trends are worrisome. This matches up with what's described in the literature and the particular studies that AHRQ has been doing for decades. And so one of the questions which naturally came up is, can we build a predictive model to predict postpartum hemorrhage? And indeed we can, but it requires having the patient-level data as well as a fairly large amount of data from multiple institutions. And so this is where the ENACT ephemeral enclaves comes in, which is a secure cloud environment, limited data sites from select ENACT sites provide data for this project, and we have the standard analytic tools that we can use. So for this particular pilot project, and since we are still in the pilot phase, there are 4 ENACT participating sites at this point, and that includes Pitt, UCSD, UT Southwestern, and Harvard. And for this particular data set, the cohort was defined by Shrine queries, and as Olga described, one query is basically a single query which describes all deliveries, and that deliveries includes PPH2. So the cohort is really all deliveries from the sites that we want. And approximately we have 500,000 patients in the enclave from these 4 sites. And the data domains are all the standard data domains that are available in the ENACT network, includes demographics, diagnosis, procedures, lab results, and medications, as well as visit types. Currently, our data format is the I2B2 tables, but we have the ability to transform this data to other formats, like, say, for example, OMOP. And it's a limited data set. So we do have timestamps of these events, which often can be very useful for certain kinds of research. So we don't obfuscate timestamps. So this is some initial analysis we did looking at some of the risk factors and comorbidities across all deliveries, which includes postpartum hemorrhage. And right now, we have done it for 3 sites. We are still sort of cleaning up the data for the 4th site. And so these bars represent 3 sites. And what's interesting is that given that these sites are really from the 4 corners of the country, now they represent the heterogeneity that we have for this particular condition. So for example, if you look at the second set of bars, which is asthma, the prevalence of asthma is pretty high at one site and much lower at the other sites. What's interesting is the tallest bars somewhere in the middle of the graph represents gestational diabetes. And we do have a fair amount of gestational, like almost like half the population at once, at actually a couple of sites, and it's much lower at a 3rd site. So you can look at this heterogeneity and do these kind of analysis in pretty great depth once the data is available in the AWS environment. And so these are the count of data that we have, the deliveries that we have from the 3 sites. You can see on the left-hand side that the prevalence of postpartum hemorrhage is very similar at about 7.5% at 2 sites, but much lower at the 3rd site. We've just started performing some machine learning on building XGBoost models, just using 15 risk factors and comorbidities. And we started by building a model at 1 site and applying it, evaluating it to a 2nd site. And that's what this table shows, that you can build models from 1 site and apply it to a 3rd, to an alternate site. And then eventually we also put all the data together, split it up into an 80-20, did training on 80 and evaluated on 20. And that shows a higher performance because we are bringing in data from all sites. So this is a pretty preliminary work at this point. And with that, I think we will come back and hopefully at some point present after we've done the entire work on this PPH data set in the enclave. But that's where we are with one of the pilot projects in the enclave. And with that, I'll hand it over to Elena.
So there are a couple of steps that folks need to go through in order to participate. So the first is at the site or the institutional level, and this is required before you can either contribute data to one of the ongoing projects or before your local investigators can request an enclave environment for their specific study. So I'm going to put the link to our website in the chat. But the first step is for you or someone else from your institution to complete our intake form. This is basically just collecting contact information so that we can share our regulatory and governance guidance and go through that process with the appropriate people, both on the ENACT side and at the site level, to both sign the data transfer agreement and then also rely on UCSD's central IRB for participating and contributing data to the enclave environment. So once that's in place, then investigators at that institution will be able to request a project-specific enclave. And we have another request form there that just asks for some additional information on the project so that it can be reviewed by the ENACT team, ensure that it is indeed a study that makes sense and is appropriate for ENACT in the enclave environment, one that can actually be done there given the study criteria. And then you'd be contacted by the ENACT enclave team to move forward from there, sign user agreements, and then The ENACT team works with all of the institutions participating to execute that data transfer based on the needs of the study, and then onboard anyone who would need access to that data for further analysis. So again, I'm just going to toss that link in here for some more information on the enclaves. And then I will also type in my email, and you can come to me with any specific questions that you don't find addressed there, and I can triage and get you in touch with the right folks. I think Nayeli will open us up for Q&A.
Yes, thank you. And thank you, Shyam and Olga, for the presentation on how to use this. One example of cases of ENACT. I have a general questions. We talk about like some limitations of the study already. You talk about some mitigation strategies as well that you started identifying. And I'm just wondering if we are thinking of creating like a repository of this. So when new teams create new enclaves or get these queries and they face kind of the same problems, they don't have to reinvent the wheel, right? They can go and try to say, oh, this happens to me, and now I can address this mitigation strategy that can be used like that. Are you creating a repository like that? And where will that be living?
Yeah, I can take that question. Yeah, that's a great question. That as we start doing these studies, now we are figuring out some of the complexities of querying the data, understanding the EHR data, and coming up with mitigation strategies specifically for certain issues like small numbers, as well as there are other reasons also. Sometimes you have to take what you have and convert it into a query, which is a decent approximation of what you want on the network. And as we, I think, go through the studies, we will create examples of these queries and mitigation strategies, and how we have solved some of these as examples. As an example, in the CKD dataset, one of the things that came up was the primary criteria used for chronic kidney disease is eGFR. And eGFR is computed using a formula from creatinine. And that formula has changed over time. And we don't know what the underlying formula is. All we see is a computed eGFR, which comes from the EMR into the data repository. So for that particular project, instead of depending on eGFR and querying it, we ended up actually using creatinine and came up with a series of queries that would approximate the level of creatinine corresponding to a particular eGFR, because that varies with both age and sex. And also the older equation varies with race. And so we were able to do that. And that's one example of a very sort of a complicated situation where we had to take what looked like a very simple query, which would be just based on some cutoff of eGFR, and turn it into multiple cutoffs at multiple age groups for creatinine. Yeah.
Thank you, Shyam. Does anybody have questions for our speakers in the group? Well, if you have any, please add to the chat. I'll have another comment on something that we've been thinking at, and we talked with some individuals around in the sites about team science, right? That this is just, these projects are not a one-person project. In your experience, right, in your group, you have this team in one of your slides. I was wondering what is the composition of the team. I mean, technical, you mentioned a technical team that is needed. So if I were to, as an investigator, to do a request an enclave, what would be my ideal team to carry out my project?
Mm-hmm. Yeah, that's another great question. Is there have been other national projects which involve analysis of EHR data. So examples of this is the N3C project, the 4CE project, which was led by Zach Kohani in Boston, the All of Us project, and now the ENACT Enclaves. And for all of these projects, what we are discovering is that typically you need a team with at least 3 skill sets. You want your clinical domain expert to define the question which is of interest. And then you need a biostatistician who sets up a study design that's appropriate to answer the question. And finally, you need an informatics person who understands the EHR data well enough that they can approximate the questions to something which is analyzable in EHR data. And also depending on the tools that you use, like in this case, if you're using the Shrine tool to do counts, how do you translate the study design and the questions into something which is executable through Shrine requires an informatics person who understands that tool. And then when you go into an enclave, you need an informatics person often to understand what the data issues are, any data quality issues, and how do you approximate, say, a particular condition with a computable phenotype. And I think in the past projects, we have noticed that like in N3C and 4CE, that having these three skill sets really moves the project along. And so that's sort of one, I think, lesson that we have learned from it. And the second lesson is that translational scientists who come from the paradigm of, set up a hypothesis and then go out and collect the data and then do the statistical analysis to figure out whether your hypothesis is supported or not. When they come from that paradigm, they requires a little bit of mind shift that needs to happen, which is the hypothesis that you have may not be answerable with EHR data. And hence, you need the ability to come up with a slew of similar hypotheses, which will be of interest to you, so that at least one or two of them can be answered through EHR data. Thank you.
Thank you, Shyam. You raised something very important, right? Like not all questions can be answered with this type of data networks. They have a specific uses, right? And I wonder if those guidance, right? And of course, there are many study designs that we can use that, but having in mind that is super important. And just as a PI, how do you envision in this landscape of translational science, which are the CTSAs or environment, right? How these ephemeral enclaves are gonna advance translational science, if you will, in your mind? How do you envision this?
Yeah, that's another great question. So more and more, we are starting to see a lot of clinical and translational scientists use data which has not been collected for research. So these include the most common example is, of course, EHR data, but there's claims data, which for a very long time has been used in epidemiological research. And there are other kinds of data which are also available, which have not been collected for research. And how you do analysis of these data sets to answer your question is a very growing field at this point. And one example is often clinical and translational scientists are looking to answer causal questions. So they want to know whether something causes something else, whether a new drug causes a new side effect. And causal questions for a while have been thought can only be answered through controlled trials, like randomized controlled trials. But about 6 months ago, JAMA published an editorial and an article which said that with the development of new statistical methods, sometimes you can answer causal questions with observational data like EHR data. So this is the first time we have a very major journal which showed that they would be okay with receiving articles which do this kind of analysis. So using modern techniques to do causal analysis of observational data, and they would receive it as equal to the traditional studies which use randomized controls studies. So they basically said they were prepared to review these papers as producing new biomedical knowledge, similar to what you would obtain from randomized controlled trials. And so this opens up really in a big way the use of these observational data sets for answering causal questions and being able to publish in the top tier journals. Now, we require some amount of education, some amount of training from the statistical world, where people have now developed these new techniques to be able to do causal analysis of these kind of data. But these are available, and as we, I think, open these up to a vast number of clinical and translational scientists, we will see a lot more research which is being done at a very high level that can go into high top tier journals and producing actually new knowledge. Right now, we're just, I think, touching the what this data are useful for. I think there's a lot more to be done.
Thank you, Shyam. That's important. Thank you for understanding it. Oh, there's a question.
Yeah.
Go ahead, please.
Sorry, Shyam, my question
Yeah.
earlier was about the IRB was more for the enclaves,
Okay.
not so
Okay.
much shrine. So
Right.
maybe the question for you and Elena and or Olga. So each time there's a new study,
Mhm.
does the hospital sending data, do they have to do a new IRB, or is it one-time blanket IRB that they'll, anytime there's a new query, data will automatically get transferred? I don't know if that
It's
question
a-
makes sense.
It is a one-time IRB.
Okay.
Basically, the parent Act IRB should cover both counts as well as transfer of data. As new sites onboard, because you definitely have to update the data use agreement and the network agreement when you onboard for participating in the enclaves. At that time, you also have to check with IRB to see whether that needs to be upgraded to allow data transfer. But once you're done, once you do it once, you're done.
Okay.
Now, locally, though, you still might have to go through a permissioning process from the hospital or the health system side. Sometimes the hospital and the health system often say, please, just tell us what are the data sets you're transferring and their volume. But that usually is something you can hash it out with the chief research informatics officer or the CMIO, and they'll tell you, okay, maybe once a month or once every few months, just give us a list of what things have been transferred.
Okay.
Otherwise, the barrier from the governance and the IRB perspective is pretty low.
Okay. Thank you.
Thank you. We have here one more comment about the IRB, that it says that if there is relying on the ENACT IRB, and if they are using their own IRB, it's up to their institutional IRB. So I think it's
Okay.
the project specific, right? I think that's the kind of
So, just wanted to mention,
Actually,
this is Mike,
yeah.
sorry.
Mike, go
Yeah, hey,
ahead.
Shyam.
Michael.
Yeah, so we have a IRB here at UCSD that you rely on, and some folks have relied on it. And if you rely on that one, yeah, you don't, it's laid out so that you don't have to do another IRB, one for each project. But Harvard, for example, or MGH elected to use their own IRB. So it's an option. You can either rely on the one we have, or your institution can decide, no, we're going to do our own. And if you do your own, then it's up to your institutional IRB as to the rules. And what we've observed is, because of the de-identification level, and because this is a very low risk to privacy because of where it is in the enclave, that generally it's been considered, exempt, or in some cases, some IRBs may even look at it and go, it's not even subject to research. But the reliance, the IRB we have here is one you can rely upon, and it does not require project-specific IRBs. So if you rely on ours, it's what Shyam was mentioning. But if you do your own, then it's up to your own institution. Like MGH, for example, their IRB will decide whether you need another one or not. I think in general, most places will agree with a blanket one for this kind of thing.
Thank you, Michael. Yeah, but it's worth a conversation at your institutional level, because each institution is different, and they might require,
Exactly.
some.
Yeah.
So just check before something else.
And that's why we wanted to be as broadly, flexible as possible. So, you don't have to rely. If you want to rely, we have one. But if you'd rather not rely, or your institution wants to do the, you use your institutional IRB, then certainly that doesn't exclude you from being able to use the enclave. Yeah.
Thank you. Yes, governance is always tricky in these cases. Thank you for that. Any other comments? No. I want to thank our speakers and welcome you for next time. And before you go, I will ask you to tell us how we did, right? It's a 2-question little poll and to make us better, right? We want to start with the right foot. There's evaluation and it's just super quick. We have also a prompt here to subscribe to our newsletter for updates, and we're gonna upcoming seminars and grand rounds and other updates in the tools. And we'll be here back in a month.